Welcome to our comprehensive guide on collecting biological data for experimental analysis. In this video, we'll walk you through the essential steps and considerations when gathering biological data, whether you're working in a laboratory, field, or clinical setting. Understanding the importance of accurate data collection is the foundation of any successful biological experiment. We'll start by defining what biological data is, why it matters, and how it forms the backbone of scientific discovery. Let's embark on this journey to unlock the secrets of life through data. Biological data refers to information collected from living organisms, including plants, animals, and microorganisms. This data can range from genetic sequences to physiological measurements, behavioral observations, and ecological interactions. The diversity of biological data reflects the complexity of life itself. By systematically collecting and analyzing this information, scientists can uncover patterns, test hypotheses, and make informed decisions in research and healthcare. Let's explore the different types of biological data you might encounter in experimental analysis. There are several main types of biological data. Quantitative data includes measurable values like enzyme activity, cell counts, or gene expression levels. Qualitative data, on the other hand, involves descriptions such as color changes, behavioral responses, or morphological traits. Both types are crucial in experimental analysis, as they provide different perspectives on biological phenomena. Understanding which type of data to collect depends on your research question and experimental design. For collecting any data, it's essential to define clear research objectives. What question are you trying to answer? Are you investigating the effect of a drug on cell growth or perhaps the behavior of animals in different environments? Clear objectives guide your data collection process, ensuring that the information you gather is relevant and meaningful. This step helps prevent wasted effort and resources, and it lays the groundwork for a successful experiment. Well-designed experiment is crucial for collecting reliable biological data. This involves selecting appropriate controls, determining sample sizes, and choosing the right methods for measurement. Consider variables that might influence your results, such as temperature, light, or genetic background. By carefully planning your experimental design, you increase the likelihood of obtaining data that accurately reflects the biological processes you're studying. Methods you use to collect biological data depend on your research objectives and the type of data needed. Techniques can range from direct observation and manual counting to advanced technologies like flow cytometry, PCR, or high-throughput sequencing. Each method has its strengths and limitations, so it's important to select the one that best fits your experimental goals. Proper training and calibration of equipment are also essential to ensure data accuracy. Sampling is a critical aspect of biological data collection. It involves deciding how many samples to collect, where to collect them, and how often. Random sampling helps reduce bias, while stratified sampling ensures representation across different groups or conditions. The choice of sampling strategy can significantly impact the reliability and generalizability of your results. Always document your sampling methods clearly for reproducibility. Ethics play a vital role in biological data collection, especially when working with animals, humans, or sensitive ecosystems. Obtain necessary approvals from ethical review boards and ensure informed consent when required. Treat all living subjects with respect and minimize harm. Ethical data collection not only protects subjects but also upholds the integrity of your research and its acceptance by the scientific community. Preparation is key to successful data collection. This includes gathering all necessary materials, calibrating instruments, and training personnel. Double-check your protocols and ensure everyone involved understands their roles. Proper preparation minimizes errors and ensures that the data you collect is consistent and reliable. Take time to review your checklist before starting the experiment. Quantitative data collection involves measuring numerical values such as concentrations, weights, or counts. Use calibrated instruments and standardized procedures to ensure accuracy. Record data systematically, using tables or digital tools to minimize transcription errors. Consistency in measurement techniques is crucial for comparing results across samples and experiments. Qualitative data focuses on descriptive information, such as color changes, behavioral patterns, or morphological features. Observations should be recorded in detail, often using photographs, sketches, or detailed notes. Consistency in how observations are made and documented is essential to ensure that qualitative data is meaningful and comparable. Digital tools have revolutionized biological data collection. Software and apps can automate data entry, reduce human error, and facilitate real-time analysis. Tools like electronic lab notebooks, data loggers, and specialized apps help streamline the process and improve data integrity. Choose digital solutions that fit your workflow and ensure data security. High-quality data is the cornerstone of reliable experimental analysis. Implement quality control measures such as duplicate measurements, calibration checks, and regular reviews of data entries. Address any inconsistencies or anomalies promptly. Maintaining data quality ensures that your results are trustworthy and reproducible. Metadata provides context for your biological data, including information about how, when, and where data was collected. Record details such as date, time, location, instrument settings, and environmental conditions. Comprehensive metadata makes your data more valuable and easier to interpret, share, and reuse in future studies. Protecting your biological data is essential, especially when dealing with sensitive or confidential information. Use secure storage solutions such as encrypted drives or cloud services with strong access controls. Regularly back up your data to prevent loss due to technical failures or accidents. Data security safeguards the integrity and privacy of your research. Effective organization of biological data makes analysis easier and more efficient. Use clear file naming conventions, logical folder structures, and consistent formats for data entry. Well-organized data is easier to locate, interpret, and share with collaborators. Invest time in setting up your data management system before you start collecting data. Missing data is a common challenge in biological experiments. Document any gaps in your data and the reasons behind them. Use appropriate statistical methods to handle missing values during analysis, such as imputation or sensitivity analysis. Transparent reporting of missing data helps maintain the credibility of your research. Validating your biological data ensures its accuracy and reliability. Cross-check entries, compare results with known standards, and use validation tools where available. Regular validation helps catch errors early and strengthens the overall quality of your dataset. Make validation a routine part of your data collection process. Many biological experiments involve teamwork. Establish clear communication channels and standardized protocols among team members. Collaborative data collection benefits from shared expertise but requires coordination to ensure consistency. Use shared digital platforms to facilitate collaboration and data sharing in real time. Collecting biological data in the field presents unique challenges and opportunities. Prepare for variable conditions, transport equipment safely, and ensure accurate recording despite environmental factors. Field data often provides valuable insights into natural behaviors and ecological interactions that can't be replicated in the lab. Laboratory settings offer controlled environments for biological data collection. Ensure all instruments are calibrated, follow standard operating procedures, and maintain a clean workspace. Lab data is often more precise, but attention to detail is crucial to avoid contamination or procedural errors. In clinical research, data collection involves working with human subjects. Prioritize patient privacy, obtain informed consent, and adhere to regulatory guidelines. Clinical data can include medical histories, test results, and observational notes. Accuracy and confidentiality are paramount in clinical settings. Proper handling of biological samples is essential for data integrity. Use appropriate containers, label samples clearly, and store them under suitable conditions. Follow biosafety protocols to protect both the samples and the researchers. Accurate sample handling prevents contamination and ensures reliable results. Efficient time management is crucial during biological data collection. Plan your schedule to accommodate sample collection, processing, and recording. Allocate time for unexpected delays or troubleshooting. Good time management ensures that data is collected at the right intervals and maintains its relevance. Sometimes, deviations from the planned protocol are unavoidable. Document any changes or unexpected events during data collection along with their reasons. Transparent documentation helps interpret results accurately and supports reproducibility in future studies. Reproducibility is a hallmark of good science. Keep detailed records of all procedures, materials, and conditions used during data collection. This allows other researchers to replicate your work and verify your findings. Reproducible data strengthens the credibility of your research. Accurate data entry is vital for reliable analysis. Enter data promptly, double-check entries for errors, and use standardized formats. Digital tools can help automate and validate data entry, reducing the risk of mistakes. Consistent data entry practices make downstream analysis smoother and more accurate. Modern biological experiments often generate large datasets. Use database management systems or specialized software to organize and analyze big data efficiently. Regularly back up large datasets and ensure they are accessible to authorized team members. Managing large datasets requires planning and robust infrastructure. Data cleaning involves identifying and correcting errors or inconsistencies in your dataset. Remove duplicate entries, correct typos, and standardize formats. Clean data is essential for accurate analysis and meaningful results. Make data cleaning a routine part of your workflow before moving to analysis. Annotating your biological data adds valuable context and meaning. Use standardized vocabularies or ontologies to describe your data, making it easier to interpret and share. Proper annotation supports data integration and comparison across different studies. Choosing the right storage solution is crucial for preserving your biological data. Options include local drives, external hard disks, and cloud-based services. Consider factors like data size, security, and accessibility when selecting a storage method. Regularly review and update your storage strategy to keep data safe and organized. Regular backups are essential to prevent data loss. Use automated backup systems and store copies in multiple locations. Test your backups periodically to ensure they can be restored if needed. A solid backup strategy protects your research from unexpected disasters. Sharing biological data with collaborators or the wider scientific community accelerates discovery. Use secure platforms and follow data sharing agreements to protect sensitive information. Proper documentation and metadata make your data more useful to others and foster productive collaborations. Be aware of data privacy regulations that may apply to your biological data, especially when working with human subjects. Regulations like GDPR or HIPAA set standards for data protection and privacy. Ensure compliance by implementing appropriate safeguards and obtaining necessary approvals. Once data collection is complete, the next step is analysis. Choose appropriate statistical methods and software tools based on your data type and research question. Careful analysis uncovers patterns, tests hypotheses, and leads to meaningful conclusions. Always interpret results in the context of your experimental design. Data visualization helps communicate your findings effectively. Use graphs, charts, and images to present results clearly. Good visualization makes complex data more accessible and highlights key trends or differences. Tailor your visuals to your audience for maximum impact. Interpreting your data involves drawing conclusions based on your analysis. Consider the limitations of your data and any potential sources of error. Relate your findings back to your original research objectives and discuss their implications for the broader field. Reporting your biological data accurately is essential for scientific progress. Follow guidelines for data presentation and transparency. Include all relevant details such as methods, sample sizes, and statistical analyses. Publishing your data in reputable journals or repositories increases its visibility and impact. Archiving your biological data ensures it remains accessible for future research. Use reliable repositories or institutional archives that support long-term storage and retrieval. Proper archiving preserves the value of your data and enables new discoveries down the line. Every data collection process offers valuable lessons. Reflect on what worked well and what could be improved in your approach. Continuous learning and adaptation help refine your methods and enhance the quality of future experiments. Be aware of common pitfalls in biological data collection, such as inconsistent measurement techniques, poor documentation, or inadequate sample sizes. Anticipating and avoiding these issues can save time and prevent costly mistakes. Learn from others' experiences to strengthen your own data collection practices. Biological data collection methods are constantly evolving with new technologies. Stay informed about the latest tools, software, and best practices in your field. Embracing innovation can improve efficiency, accuracy, and the scope of your research. Proper training is essential for anyone involved in biological data collection. Invest in ongoing education and skill development for yourself and your team. Well-trained personnel are more likely to collect high-quality data and adapt to new challenges as they arise. Collaboration enhances the quality and impact of biological data collection. Working with experts from different fields brings new perspectives and skills to your research. Foster a collaborative environment to share knowledge, solve problems, and achieve common goals. Collecting biological data for experimental analysis is a multifaceted process that requires careful planning, attention to detail, and ongoing learning. By following best practices in data collection, organization, and analysis, you can generate reliable results that advance scientific understanding. Remember, the quality of your data shapes the impact of your research. Thank you for joining us on this journey through the world of biological data collection.